R and Bioconductor. Oh, don't worry. And yeah, so just to start, for this workshop, I prepared this actually image TCGA workflow. So all these packages are the base name of image TCGA. Image because we work with images, and TCGA because we work with public data from the Cancer Genome Atlas. So on this link, you can find the website, and then we can just follow all together. Maybe I can send on Zulip later, so we can also follow by the website. So that's more or less the structure of the packages. So the starting point, like here, there are the histopathological images. Then we use image TCGA. That image TCGA, these are Bioconductor package that create a Shiny app in which you can explore the HND images. And then there are three different packages that are image feature TCGA, image TCGA utils, and histoplot-R that allows you to deal with this kind of data. And the last one is the one in which we can give information about the downstream analysis. So that's the structure of the repo. When you will be able to open your RStudio session, you will have a R folder that is named image TCGA workflow, like this one. And inside we have like the R folder, like in the R package, but this R folder is empty. And we have in the vignettes a lot of R Markdown file. There is one specific for today's lab, but I also give you more R Markdown if you want to maybe understand more some stuff or want to do other analysis. Maybe not today because we don't have so much time. And then there are other information, but that's the most important things. So for today, the first 15 minutes will be like a sort of introduction. Then the second part will be the introduction of the TCGA resources and the 4 packages. And then we deal with the packages. The first one will be image TCGA. Then we explore the HoverNet output that is part of the image TCGA project. Then we will explore the ProfGigaPath output. And then we try to make all together with a MOFA model, like it's a multi-omic factor analysis model, and then a question and answer. So starting with the first part, for today, we work with the TCGA images. There are more than 11,000 images from 32 different tumor types. They are all saved in this SVS format. This format is typical from the previous scanner. So if you use different scanner, you have different file format, and sometimes you have to convert maybe from TIFF to SVS or from SVS to TIFF or other one. To download all these images, we took like 25 days. That is around 11 terabytes of data, but I just say that we don't do this today. And so these diagnostic slides are important because the resolution that these images provide is the higher possible for the TCGA data, and they were used for the diagnostic purpose. Also on the TCGA, there are available other images that are at 20x resolution, but they are more used maybe to be sure that the tumor that you are analyzing is actually tumor and not just a normal sample. So for the one who are not confident with the digital pathology is becoming very routine right now during these days. And because there are a lot of new models that use the GPU, also foundation model, convolutional neural network models. And with this model, the idea is to try to improve the patient care and also support the pathologists in their work. So histology is the study of the normal tissue, and pathology is the study of the disease tissue. So histology plus pathology make histopathology, so the name that you usually know about it. And HND is the stain in which you can find these images like the atoxylin and the eosin 1. So for example, with the purple, you can see the nuclei, and with the pink one is all around. So, for example, what do you see? Which organ, healthy or sick? For example, I'm not a pathologist, so I'm not able to say anything about these images except for the shade of the color. But what a pathologist sees here, the pathology, for example, here, you can see like healthy lung tissue that there are blood or sacs of air. And here there is a diseased lung tissue with tight cell and bloating nuclei. But also we can say that because a pathologist annotate these images, but also sometimes we can say also this information using this kind of model like foundation models or convolutional neural network models. So these images are huge. And for example, this is like an HND images. This is an image that you can take with your phone. So the size is completely different, and also the resolution. And so when you talk about HND images, and they are very huge, you talk about these whole slide images, also called WSY. And the size of these OSLite images can range from 500 megabytes to 5 gigabytes. Some now also more than 5. You can find also about 10 gigabytes, but they are just the outlier of the distribution. So when you work with these images, the smallest measure of unit is the pixel. And then you talk about resolution. This is a particular term that you use to describe the crispness of an image. And the higher is the resolution, the number of pixels will be higher, and also the quality of the images will be better, and you're able to detach more information. So as I said before, we have this image TCGA Word. This one is the first package, image TCGA, and this is a Shiny app. And now we can run in R and see how to interact with these images from the TCGA. You can see where they came from. And also in the next release of Bioconductor, you are also able to plot like survival curves, like Kaplan-Meier from the images that you select. And then also there is image feature TCGA, in which we make available all the feature that we pre-computed on this data, like the TCGA one. And image TCG utils is a package that helps us to deal with this kind of huge data and the feature that we extract from the images. So image TCGA again is a Shiny app. Image feature TCGA, for example, help us to analyze and have in our computer the pre-compute image feature. You can use like get catalog here with HoverNet and get catalog with Prof Gigapuff. And then image TCGA utils, that is very helpful if you have maybe to deal with these two kind of output. So you can also merge HoverNet and Prof Gigapuff output on the same slide. So first step, now it's because we run on these 11,000 images, two model, Prof Gigapuff and HoverNet. And then we saved the result and make them available on these two packages. And so instead of running for 2 years the same model, you can just download the packages in a couple of minutes. And so now if everything is working with your RStudio, yes, okay, we can move to the RStudio server session. And I don't know if you have the same, you can see, I can zoom it a little bit. Quite better? Kind of. Okay, so here you have this image TCGA workflow folder. And then inside, as I told you before, there is this folder, vignettes. And then we just have to take the workshop EuroBioC 2026. And also here on image TCGA workflow, you have the render version of the R Markdown file. So you can also follow from here. I send them Zulip, so you are able to follow. Oh, image TCGA workflow. Okay, yeah. It's working. You joined the Bioconductor workspace before? Okay. Yeah, there is this workshop instruction. They put it on Zulip. Okay, and if you follow it, okay. There is no channel, right? On Zulip for the workshop. In the general channel. Maybe I can go. Okay, so on Zulip, you should have the link right now. Okay. Oh, no. So many. Okay, now it's fine. Sorry. Okay. Yay. So let's go to R. And yeah, we start right now. So again, the workshop description. So yeah, of course, if you have R basic familiarity, or if you have any question about how to use R or Bioconductor packages, is the first time that you saw them, just let us know. Also, my lab mates will help you if you have any question. So just raise your hand. So let's see. So if you go to line 149, we are just load the first library, that is image-TCGA. It works. Okay. Oh, yeah. Oh, yeah. Oh, easy Zoom. Yeah, it's not, because I'm not able to connect it to the projector.
Okay. Full screen will be better. At the bottom, full screen. Is it better now? Yeah.
Okay. You can see, right? Okay. So if you load the first library, then in this package, there is just one function that is image-TCGA, because this package is just for our Shiny app. And if you run, they should open a new web page, like a new interface with a Shiny app, in which you can just start in explorate different projects, different sample types or information. So as I said before, we have 11,000 images. They are from 9,640 patients. And then you can, for example, just filter for the ovarian cancer. And then you can see that in the number of the images and the patient will change. Then also you can add other tumor types and see how much the number changes. And then there are different panels, for example, like the dot plot of the geographic distribution. And in the dot plot, you can also select the X and Y parameters and the coordinate and what you want to see. For example, instead of the project ID, maybe actually on the Y-axis, instead of the sample type, we can maybe want to visualize the city. So you can see where all these, the Cancer Genome Atlas images, where came from. There is also a very interesting question about these images, because they talk about a lot about batch effect, about images, about different scanner. So having this information to analyze this kind of data is quite important, because they give us a lot of proxy information related about how they process the images. Yeah. And also you can play here like a playground, and yeah, for example, you can also visualize different state or different continent. So it's up to you. And then we have also the geographic distribution, which again, you can visualize. We can add more filters. So remove images and just visualizing the ovarian cancer, that we can see there the majority part from the US. And then you can also zoom in and see from which hospital or labs they came from. Yeah, I don't know if you want to just try it. Also on the first panel, the summary statistic here, you have like this generate our code, in which you can just, for example, now we have 106 patients, 107 images. If you click to generate our code, here you have all the R code that you have just to copy and paste on your R session to download the raw images on your computer, but don't do that because each image is about 2 gigabytes. So it takes a while, but yeah. Then just closing the Shiny app, your R markdown session will start again and you can start working again. So yeah, if you have any question or comments. Okay. Okay. Yeah, here, for example, you can use the library Genomics Data Commons. That was the same as in the Shiny app. And then you can, for example, if you want to download these images, that will be the example that we use for all this lab. You can also download it. You just make uncomment the line 117 and 71, and then you will be able to download the images on your computer, but that would be about 2 gigabytes. So don't do it right now, maybe later if you want. Okay, so for the third part, we are going back to the slide. And we talk a little bit about quantitative pathology. So when you have these raw images, they are just colors, but then for integrate with analysis, you need to transform these images in actually numbers. And there are two kinds of ways to do that. There are the convolutional neural networks and the foundation models. And for right now, we just focus on the convolutional neural networks. And both for foundation and convolutional have different tasks. For example, the feature extraction. So you just extract some numbers, a sort of dimensionality reduction, like the PCA, the principal component analysis. Then you can perform nuclei segmentation. So you're able to have the contour of each nuclei, the nuclei classification indeed. So you can say if it's tumoral, non-tumoral, stromal, necrotic. And then also there are new models in which you can try to infer the RNA expression from the images, or try also to infer the spatial expression from the images. But right now the model need to be well trained if you want to do that. And also if you want to do like try to infer RNA expression, maybe try with some images that also have the omics data. Because one of the problem of the TCGA1 is that you have the images, but we don't have the spatial resolution. We just have the RNA-seq data, like the bulk, and the methylation, mutation, proteomics, but we don't have the spatial level. So the convolutional neural network, as the name said, are based on this neural network. They are just like interconnection of nodes that are these neurons, and is organized in layer. So more or less, it's a structure that happen in our brain. So we have like the neurons that were activate or deactivate, and from some things that happen in biology in our brain. And this model is able to learn from data that they perform some task, for example, classification, regression, or pattern recognition. So that's more a kind of story about these, where this neural network came from. So they are kind of old because they are from 1943, but this was the first time in which they had this idea about neural networks, but they don't have the resources and the mathematical knowledge to make these neural networks work. So years later, like 5 years ago, 10 years ago, they start again work with neural networks. And there was the perceptron neural network that is the easiest one. So we have just one layer for the input and one layer for the output. And then they start to create, okay, maybe at some point we can also add this hidden layer in the middle. That is another neurons that were activated by this edge here. And then we have the fully connected neural network in which each neurons is connected with the edge to the previous one and the next one. Again, we have an input layer and an output layer. So this is the more complicated, fully connected neural network. When you talk about the images, you talk about this convolutional neural network. Why we talk about this convolutional? Because these convolutional neural networks are sparsely connected networks. Why we talk about sparsely? Because when you have like an images, you have a lot of pixels inside, and here you are able to see that it's green. It's like a tree, and the 3 is green. So if I took a pixel here, is green, but the next one is like a shades of green again. So the correlation about the 2 pixels is very high. So we don't need both the pixel to say that is green or part of a tree. But so more or less is the main reason that the convolutional neural network works, because you don't need the whole amount of pixels, but just part of them. And when you have like a convolutional neural network, you have your input layer, like the original images. And then in this convolutional here, like in this small square, you are like try to summarize the information that is on this 3-4-3 matrix. With this GIF, it's more easy to understand because the convolutional layer enter on the matrix and just with these 3-4-3 matrix, just go right and then down and just try to summarize the information in a small matrix. So when you deal with images, you have this convolutional layer in which you are going to summarize the information, then the pooling layer in which you are summarize again the information in into the convolutional, and then you have to going back to a 1D vector because you have to do like regression or classification, and then it will be the input of the fully connected neural network. That's a cute video of like this lasagna maker, like lasagna, and you give an input a dog, and then you divide your lasagna dog into 2 different images, and then here you are still able to see there are 2 dogs. That's why convolutional neural network works. But then if you take these 2 dogs and go inside again the pasta maker, at the end we have 4 dogs. But let me know. I'm able to see there are 4 dogs, but just give me your impression because. So actually we don't need all the pixels in the images. We just need part of them to understand that it's a dog. And it's more or less the same with biological images, maybe to understand that it's tumor or a stromal or something else. Okay, then the pooling layer is going to summarize again the information inside the images. And there are 2 types of pooling layer. For example, this one is the max pool. It's going to take like the maximum value in your square. So in the blue, in your square. In the blue one, it takes the 9. In the green, the 7. In the yellow, the 8. And in the red, the 6. But then if we use another methods like the average pool, from the blue and the green and the yellow, we have the same number, that is 4.25, because we are just doing the mean value across the 4 number, not to taking the maximum. So depends on these the methods that you choose for the pooling layer, you can have different results or summarize the information a different way. And then again, this one direction vector in which that you need to be like the first input for the fully connected layers. Okay, so then I'm not going into this kind of stuff today, but that's kind of interesting that how works the learning process of a neural network works. You start here with the forward propagation. That is the first step in which you have a sort of prediction value in which you are going to compare with the loss function. Remember that you want to minimize your loss function. You want this loss function is close to 0. So what does it mean? That your model predicts well. And with your loss function is again an input for the optimizer. And with this optimizer, that could be like a gradient descent or something like that, in which you are going to find the function that maximize or minimize your value. And then you create new weights and you start again with the neural network, again with the forward propagation, then the loss function, then again the optimizer. And you are like stuck in this loop until you have very good prediction. And each loop is called epochs. Again, we have another neural network playground. So for example, just saying a number, 1, 2, 3, 2, 40, no, just 4, okay. If I'm going to draw here, it's the original layer, like the input layer, and you can also hide this one. And then we have all the convolutional layer and the downsampling layer. They are going to summarize the information and divide into different layers. And here we have the fully connect layer. That is the one that will be the input of the convolutional neural networks. And then this one is our output layer. So here, for example, the first guess is 6. So it didn't work. Okay, that's funny. And the second guess were 2. But maybe because that's my way in which I draw a 4. But maybe if I draw like that. Yeah. It's guessing right now because depends the way in which you draw the number, because probably this model is trained on this kind of 4. But if I draw my 4 in my way, it is not able to recognize. So maybe it need to be retrained again. Okay. So this ResNet 50 is one of the convolutional neural networks that are used a lot with image analysis. And if you read a paper about the image analysis and convolutional network, if they don't cite this paper, it will be weird or maybe very revolutionary. And this kind of model is also part of overnet. Overnet is the model in which we are going to understand the output in a couple of minutes in R. And this overnet is, again, a convolutional network and give us 3 different outputs. The first one is the nuclear pixel branch, this here, in which the output is like a mask, in which you have this binary situation, like black and white, in which you have the tissue and the area with no tissue. And then we have the hover branch, because hover means horizontal and vertical one. So we are going to create 2 different maps in which they, with the sort of mathematical stuff inside, they are going to know the size of each nuclei and the coordinates of each nuclei. And then there is the nuclear classification branch, in which we have information about the cell type of the nuclei, like stromal, benign, necrotic, inflammatory. So these images is made with R, with the output that we got from overnet, and this one is the raw images. And yeah, so now we are going back to R. And as you wish, I can just show you the code, or maybe you can just want to try to run the code, and if you have any question, just let me know. Maybe I can show you. I don't know. Okay, for example, of course, we need to load the library before. So we need to import the image feature TCGA, is the image plot. Also, we need to load spatial experiment, because this kind of information, like the position of each nuclei, you can store them inside of a spatial experiment or a spatial feature experiment object. Remember that the counts level, like the counts metrics in this object, will be empty, because we don't have a spatial omic like a gene counts for this kind of images. But we need the spatial experiment structure. And then dplyr or ggplot to deal with graphical, and also we need to tie our data. For example, the output from overnet could be like a JSON file. And in the JSON file, we have the coordinates of each nuclei. And also in this data, we have also the probability of each for each nuclei. So we know, for example, the probability of is tumoral or no tumoral. And then we have this H5AD object, which we have also the same information as the JSON, but also we have pre-computed other feature that you can find inside the H5AD object. So with get catalog, if you run this first command here, then you have to write here yes, because this package is using BioC5 cache, that is a Bioconductor package to deal with the cache in your R session. So you have to say yes. So it creates a sort of folder. And it start downloading stuff. So the first time that you download stuff with BioC5 cache, it require maybe more time. But the second time, you just read your file from your cache and will be faster. Okay. So we are going back here. And then we want to explore the catalog for overnet. it would be a long metric, so we are not going to analyze everything. But we have, for example, from overnet, we have different format like the JSON or the GeoJSON or the H5AD or the PNG, because also we have the low resolution format for this file. And then we have all the file name and the full path, that is the folder in which they are localized. And then we have also other information like the file name, like the complete file name, that it had to deal with the curated TCGA packages, so deal with the TCGA data. And then all the same information that you can find in the image TCGA Shiny app. So the city, the country, also the latitude and longitude, just to make your a dot in your country, like in the map. Okay. It's working now in your R? Okay. Okay. So that would be tricky because, let's see if it works. Because before we go out of memory, so we require more RAM on these RStudio sessions. So if they manage, we make it. If not, I have some R data that I can share with you, and it will be easy to deal.
Try to give you a bit more RAM. Now it's 20.
It's 20. It should be fine. Yeah, before it was that at 14.
Oh.
So let's see.
Good question.
Yes.
You have your own data popping it into this model. Is there any sort of pre-processing that has to be done? Like, so I work in mammograms. So
yeah.
There's a lot of reshaping, stratifying.
Yeah.
Same thing apply here?
Yeah, so here in the image TCGA workflow, we prepare a lot of information, for example, like data access, how we download the TCGA diagnostic. Also, if you want to reproduce the overnet extraction, if you go here, we have all the templates and information about what we need to run this model. For example, for the GPU, we need like NVIDIA A100 or L48S. They are like huge GPU. And the RAM, we need 60 gigabytes of RAM. And at the end, the storage for all these overnet output is around 780 gigabytes. That it's a lot. And for processing these images, we took like around 1 hour, 1 hour and a half. Depends about the quality of the image itself and also the size. And yeah, if you run again, this model are pre-trained on raw images, so we can give in input like raw images, but you can also perform on these images like normalization or color correction, or maybe sometime on your images there are like the bubble or something, because that's like a technical problems from the scanner, and there are some tools that are going to remove this kind of problem. But actually with overnet we don't need to do that because it works very well and clean also the images. As the same for Prof Gigapath, that is the second model that I show you later. No, okay, we go out of memory. So yeah, but I have our data, don't worry. Yeah, maybe I just show you. So from the get catalog from overnet, you can just filter. For example, we want to work with these particular images that we choose randomly from the ovarian cancer. And the output is JSON and gzip because it's a zipped file just for the memory issue. And then you have the function get file URLs, because all these outputs are saved in a bucket, like an NIH bucket, and you can have access to a dedicated URL link. And then here with the overnet, we specify that is an overnet output, and the output class in our case that we want is spatial experiment. For example, if I just change here spatial experiment with spatial feature experiment, in that case, your new object in R will be a spatial feature experiment. Yeah, for example, yeah, to run again the library. So instead of going in, like putting input the JSON file, we can use the H5AD in gzip, and in this case, it shouldn't go out of memory. It's quite fast actually, because that's why it's the way in which the file are created, because H5AD is like an undata object with the H5 structure. Undata is very used in Python, in ScanPy or Rapid single cell, but also right now in R, there are a lot of packages to deal with undata object like undata R or others. And so the H5AD object, it's worked quite better, because here it just requires 1 GB of RAM, but using the JSON file, we went out of memory. So let's see, maybe something that we have to fix in the package, of course. And if we go on to explore this type of output, we have, as I said before, we have a spatial experiment. In this case, the dimension here, it's so the dimension is 1 because we have just 1 images, and this number is the number of nuclei that are detected from overnet in the original images. On the assay, we have X that is created by default from undata, and for the mean intensity and this neighbor distance, they are just computed with some SquidPy or in Python, and then make available through R. In the cold data, we can just explore the cold data. You can see there, yeah. The cold data of yeah, the name is overnet spatial experiment underscore H5AD. Just we are on the same page. You can see here that in the cold data, we have like a list of data, and we have like the type. So it's a factor variable. And if we check inside, we have like neoplastic, stromal, inflammatory, necrotic, that are all the cell type provided from overnet. And then we have also the mean intensity, maximum intensity. They are information based on the color in your images. And since there, you can just go inside the spatial experiment, explore, see how many nuclei, if the distribution of the nuclei is the same across all the cell type. You can just perform some quality control analysis. Here, with the TAMPAF, we are just going to retrieve the PNG images. It's like a raw, low resolution images. And it's helpful when we want to plot these overnet images on the original one. And again, it takes a while because you have to run, but yeah, it should be fine. Yes. Yeah, for example, this is the images. So this one is the raw images, and then this is the images color. Actually, is the same images recreated with the overnet output, and you have a lot of color like benign, inflammatory, necrotic, again, all the cell type. Okay, let's go to Prof Gigapuff. So speaking about quantitative pathology, there are two ways, like the convolutional neural network and this foundation model. That's my understanding of convolutional neural network and foundation models. So when I read a paper, people tend to present like this foundation model, like these very powerful Pokemon like Charizard, of course. And but also I want to say that without Charmander, you cannot have Charizard because it's like his evolution. So when you deal with the convolutional neural network, they are real powerful too, not just because now it's very catchy to talk about foundational models. So yeah. So just to give you a small introduction about foundation models that are like these huge models that are trained on board data, and usually with these self-supervised learning, and they can be adapted to a wide range of downstream tasks. Actually, the most easy way to describe these foundation models is that they are like models that are trained on billions of data, and they perform very well. Actually, they require a lot of GPU, like graphical process unit, and the cost, the training part could be very long and cost a lot of resources. So in foundation model, there are a lot of foundation model, but the one in which you are more interested in the image project is the vision transformer, in which are going to use the information pre-trained on a lot of images like the HND1, and through a self-attention and convolutional process tends to give you a embeddings matrix. What is this embedding matrix? It's just sort of like dimensionality reduction matrix, but it's not PCA because it's not like a linear encoder autoencoder situation. It's more complicated, but the concept is similar. So there are some parallelism when we use like ChatGPT or one of the LLM model. We give an input this sentence, and each word in the sentence is called token. And then we have through this transformer, like with a self-attention process, the output is a sort of probability of having a word after another one. So if we move to the vision concept, the sentence, when you write something, is the image. And then each token, like each word, is this patch. So if you have an original images, you can just cut these images into small pieces, like a small square, and each square is considered like one of the word of your sentence. And then again, you have for each of these small square a sort of value, like a vector that give you some information transformed in number. That's important because when you talk about a sentence, each word has a specific meaning, but this meaning, like station, change based on the context. So you can have station like the tray one, or the one in the sky, or like the one for the radio. So there are different kind of station. It's the same when we talk about this patch, because if in this patch we have like a that was funny. If in this patch we have like a necrotic cell or something like that, is change if this necrotic cell is surrounded by stromal cell or maybe inflammatory cell. So the context also in biology matter. So again, this transformer read into these token or patches and the context, it's really important. So the model that we use for the HND's images is Prof Gigapuff and was trained on a billion of images like this one that were cut into different tiles. You can talk about tiles when you say that you are just cutting the images, so just small pieces. But when you refer to models like AI, like the foundation model, tiles and patches are synonymous. So, but patches is more used in the AI context, tiles in a more general one. In this model, there is something very interesting that is the knowledge distillation. And this one was proposed by Geoffrey Hinton in the 2015. And here you have like the teacher model and the student model. So inside this foundation model, you have this kind of relationship across these two models, because the teacher model was trained on billions of tons of data, and the student model is just trained on the output of the teacher model. So as if you are like a PhD student with your supervisor study a lot, learn a lot of stuff, then you have your weekly meeting, and then you took like the output from your supervisor, like suggestion. And with this information, you go to study again, try to go into your field, and then you go back to your supervisor the next week with new information. And with this kind of new information, you are trying to train again your teacher. So this is a kind of loop, and this is really important, is kind of this foundation model, because the learning of the teacher require a lot of time, but this relationship can also be very helpful to make this model go run faster and without so many resources. The output of Prof GigaPath could be like tile level or slide level. When you have a tile level, you have these metrics like this one, and the tile embeddings are from 1 to 1,536. And then you have the tile. So the tile is one small patches into the images. And then when you have the slide level embeddings, each row is a different images, and the number of column is different from 1 to 768. So that's something that I'm going to do now in R. So we have our raw images, the overnet output, and then this is the Prof GigaPath output. So we're just going to compute the principal component analysis of our slide level embeddings. So on these metrics here, on these metrics here, like the tile level embeddings. And then we are going to reconstruct these images without the real images, but with Prof GigaPath. And you can see also that the color of the loadings of the PC1 tends to represent the same information provided by Overnet. And no, that was just interesting because we just compute the UMAP on all the images provided by the TCGA, and you can see they tend to cluster. And that's very interesting because what does it mean? That the different tumor types has similar information that Prof GigaPath is able to detect. And then, yeah, now it's the time to go more into these results and try to understand more biology. Yeah. Yeah, for doing that, we can use PRCom. That is just one function. So, yeah, again, we have the get catalog. And we can have all the information about Prof GigaPath, just typing Prof GigaPath. And then we can just detach the information from Prof GigaPath for the slide and for the tile. As before, here, we have to add one layer because we have to specify the level if we want the slide one or the tile one. And then, again, with Prof GigaPath, we are just going to create the way in which we are going to have this data. Yeah, no, I just show you with R, it's more easy, sorry, to understand, yeah. Yeah, with this kind of data, it's more easy because the size of the matrix is lower, it's smaller than the one in the J zone or the H5AD, because in that case, we have like millions of nuclei, but in this case, we are like thousands of tiles, or in the case of the slide level, we have just one vector for each images. Yeah, so we are going to extract the information in the embeddings. If it's one. Yeah, so you see here that our Prof GigaPath tiles has this size, like 4,261 observation. That is the number of the tiles in which the original images is divided into. And then 1,543 is the number of the embeddings provided by the model. So for each images, we have the same number of columns. Okay, and then with these images, we are going to compute the PCA. So right now we are just saving our embedding columns here. And maybe I just show you something. Yeah, for each images. Yeah. That's the structure. So each row is one tiles. So the name of the images is actually the same. So this first column is not so helpful. but then it's very helpful combined with the tile ID. So it's the number of the tile in which these original images is divided into. Then in the tile name, we have also the information about the X and Y coordinates that help us to reconstruct our images. And then from 0 to 1,500, whatever, we have the number of the embeddings. And on these metrics, we are going to compute the PCA because 1,500 columns, there are a lot of information and we are not able to understand anything. So we are going to summarize with the PCA and with 50, 25 PCA. So the resolution will be slower and it's just a way to summarize your data. And then from here, we can just recreate our just embedding calls. That is just an object that contains actually just a vector with the name of the columns. And then in coords, we are going to save the information about the coordinates, like the X and Y. And then to compute the PCA, we're using PRComp. Just considering the columns that are numerical, like the embeddings. That's why we did that, because if not, we just also have the information about the X and Y and the name, and we don't want to use them in the PCA. And then just for a graphical point of view, we just save the result of the PCA on this data frame. And then with the ggplot, we're able to reconstruct the original images with the color of the PC. That's quite interesting, also going to see how the things changes using maybe PC2, PC3, PC4, because also, not all the variability is explained in the PC1, but the majority of part, yes, of course. I answer, yeah. Okay. Yeah.
Is there any advantage that is known just in benchmarking
or okay.
In the future?
Actually, there are a lot of foundation models right now for HND images. Prof GigaPath is just one of the examples. Another models are, for example, UNI, Conch, or histopath. There are several ones. And for UNI, they made on GitHub repo a benchmark. Maybe I add in the final slide. Yeah, this one. Yeah, here, for example, they use a different pre-trained model, for example, for the vision or the language. So they use a model trained on language to predict information from the images. And actually, they're not performed very well, and makes sense, but they also try to do that. And actually, I guess my personal opinion is that foundation model and convolutional neural network have a different task. Because with a convolutional neural network, you are able to understand more pattern from the images. You can do segmentation and classification. And right now, with these embeddings, of course, you can also perform a classification, but the the architecture of the model it's more complicated. And if I have to say, maybe sometime using something simple like a convolutional network could be not the best, but maybe I want to give them a chance to this model just to see if they perform well. And but I guess the foundation models part is that they are pre-trained of a lot of information, a lot of different images, but sometimes maybe there are too many. I don't know. There is a limit of images and number to pre-train, because at some point the information can be redundant, or also there can be biases, because as you see from the number before, if you train on the wrong data, it will predict not so good like the 4. So yeah. And also, I guess that right now the information about the pathologies like external notation are really important, because they give us like a sort of gold standard, or kind of like a benchmark of if we are doing good or wrong, of course. And yeah, and going back to R, then we have the point pattern analysis. And this it's really interesting because it's one of the analyses that you can do on this point. So if you consider every cell like a point, you can try going to understand if this point tends to cluster together, if there are some correlation between adjacent nuclei. And it's in terms of biology, it's really interesting when you study, for example, tumor microenvironment or cell-cell relationship, and you can perform these metrics. For example, this is local Morans, and we compute for the PC1. And again, with these metrics, we can see how different cell nuclei tends to cluster all together. And here you can see that there is a high level area with these metrics that is high. So maybe in this area, there is a not so high variability in the data. And then just to conclude with the slides, and then we can just do stuff in R. On the 7th part of the R Markdown, we have this MOFA, that is multi-omic factor analysis. That is on a kind of model that is really helpful when you deal with different type of omics or different type of input data. For example, with images, proteomics, RNA-seq like bulk, and it's going to compute a factor value, and just to see if different data tends to give you more information than the others. I guess these images is more explain better, because with this MOFA, the output is that you have this factor, and for example, the factor 1 tends to express more the information contained in the embeddings, and the factor 2 tend to express more the information in the RNA-seq. But then with this factor, you can play again and maybe to perform some survival analysis using the information contained in the factor, or just use this data to go ahead with your analysis. And yeah, that's it. But if we're going back to R, yeah, we can. I don't know if you want just to run R or I run and then we comment all together as you wish. I can run. Okay. I don't know. I run. Okay. 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 So that's another way in which we can import the information from overnet. Again, the input is a GH5AD object, and the output is again gzipped. And the way in which we store the data in R is using this spatial feature experiment. Again, we have to wait a while to import this data. More than a while. Okay, now it's working. Yeah, you can see that is the same information that we have before, but the class is no more spatial experiment, but this is spatial feature experiment. On the assay, again, we have the mean intensity, the neighbor distance, and in the call data, we have the type, the mean intensity, the near neighbor's distance again, the sample ID. And then in the spatial core, we have the information about the centroid of the X and Y coordinates. When we perform like this Morans 1 is, as I said before, tend to quantify the degree in which they tend to cluster all together. So it's a matrix in which you are going to compute the similarity across these cell. And for example, a positive Moran first indicate that we have a spatial clustering, while negative value indicate that we don't have spatial clustering, but like spatial dispersion. Here we are just going to match information that we have in the prob gigapath tiles with the X and Y coordinate and the PC1 and PC2, so just to have everything on the same object. And in course, we are going just to extract the information from the tile X and Y. Oh, no. You just missed something. Okay, so these two metrics then to need to compute the Moran test, because for the first one we are going to create a KNN object with k equal to 6, and then here we are going to compute the Moran test and the Giri test again for the PC1, and then Lisa again. Yeah, so here we have just the Moran test, in which you can see that one of the important things is just to check the p-value. And in this case, it's lower than 0.05. So it said that our cells tend to cluster all together, but that's more like a global metrics. But you can also have this information for each nucleic cell in your data. And then we can also going to plot this more into a scatter plot. And then we have the value of the PC1, so like the loadings. And on the Y lab, we have like the spatial lag. So we have how these cells tend to be spatially aware. So that's very confused because there are a lot of dots, of course. But if they stand on the diagonal, means that the information has a linear trend. So that's a good point. But there are some points that are out of these linear trends that are going. Maybe they are not tend to cluster. Again, speaking about this point pattern analysis, there are these Ripley KL function and the cross K function. And this first one said us it's like a test to check the clustering versus the regularity at multiple spatial scales. And the cross type K function is like a spatial interaction between pairs of cell types. For example, in our case, we just select neoplastic versus inflammatory, but you can also go into check necrotic versus inflammatory or all the one available. For this kind of metrics, you can use the SPAT-STAT package. Oh yeah, we don't have this data because we use another one, because it's the one that is not working before, but it should work again with this one. Maybe we can just. Okay. No, okay. So we need other type of data. Oh yes, of course, here on this link, you can find all the RDS object to make your analysis if it takes too much time to load. I can also share here. No, it takes a while. Yeah, exactly. Yeah, probably yes, but But here.
Maybe you can refresh the page if
No.
it doesn't work.
Okay, no, okay. Cancel. Okay, I just. Just keep that part. I want to just show you this one. If you start from the overnet spatial experiment from H5AD to the Prof GigaPath tiles, you can merge the information from Prof GigaPath and overnet on the same object. And so we just need to, again, extract this information and save this information in cell data. Oh yeah, for doing that. It takes a while to plot because I'm going to plot each nuclei in the images. Yeah, it's here. So I don't know if you can see, but the two images, like the blue is overnet and the red is Prof GigaPath, they are not matching perfectly. So with this match overnet to tiles function, we are going to match the. Oh, I will miss all the libraries. Okay, we are going to match all the data on the same images. It takes a while because inside is going to find like a scale factor to resize the two images and makes them on the same level. So now, for example, we are able to say, yeah, it takes a while. Yeah, for example, no, the resolution is terrible, but trust me, you're able to see that each dot now has a proper size that depends on the number of the cell in each tiles. So the tiles are from Prof GigaPath, like the embeddings, and now you can correlate this information with the number of the nuclei detected from overnet on the same images. And yes. And then for the multimodal integration, we can use MOFA, that is this package. And actually, we need this data that we have to upload. Okay. Okay, here in comment, you have all the code to reproduce the things that I just done. So we are going to have like an embedding matrix, RNA matrix, and a methylation matrix. The embedding matrix is the one from Prof GigaPath, and the RNA matrix and the methylation matrix are from curated TCGA. That is another bioconductor package that help you to have in your R session the data available with the TCGA. And for doing that, you just have to recall a function in which you are going to download all this data. Since this takes a while, I just saved the output on this RDS file. And maybe, nope, just. Takes a while. Yeah. No, there are no stupid questions.
It's like after you have gone through the process of converting the images into some sort of tabular data, so there's more
Yeah.
like a spatial omics data. So that
Yeah.
would mean that at that point, it's most of the spatial omics methodologies can be applied.
Yeah, no, in that case, if you have also in your images a spatial resolution, you can integrate the information. So for example, if you have your spot, you know how many genes or the expression of your gene for each spot. In that case, you can match your spot with the ties from Prof GigaPath and then correlate the gene expression with the embeddings. So yeah, you can do that. But with the TCGA one, there is no spatial resolution at all. We are trying also to simulate them using some foundation models, but we are not sure about the result because also give us expression where there is no tumor or tissue. So that's kind of weird and not trustable. But yeah, you can do that if you have your own data. Yeah.
Trying to come up with factors based on embeddings. Embeddings is already a high, like low dimensional representation, and then we are trying to get another low dimensional representation from that. Something that people do all the time.
Yeah, you can do that because in this case you're going to add more information because you're not just analyzing your embeddings from Prof GigaPath. We are just to see if the information contained in the embeddings is also inside the bulk data or the methylation. But yeah, so you can do that because they are just trying to see if they give you different information. So, you know, and then now we have the three objects. So for example, for doing that, from the 107 images, we have just 59 samples because we have to match both methylation, bulk expression, and images. And the embeddings dimension is 768. The number of RNA-seq genes is 1,000 because I selected the most variable genes just to make the matrix a little bit smaller. And then we have the methylation probes that is 23,367. Then here you have all the code to arrive on the same input data. And then with the MOFA, you have to create like a list of objects. And then giving input to this create MOFA function, in which it's just going to create this MOFA object. That is the number of the views of your data, that is 3. But also you can, if you have, you can also add more information like protein or other stuff. And then we can have a plot overview of the data. Just to see, for example, how many columns each data has. For example, RNA-seq has 1,000, methylation 22,000, and image 768. For 59 sample. If we sometime you can also keep a sample that doesn't have all the three data, but in this case you have like a vertical line that means not available or something like that. But in this case it's just everything on the same line. And then with the get default model option, I just keep the default model 1 for right now, but you can just change it as you wish. And you can set a seed for the training, the maximum number for the iteration, and then for the convergence mode, so how many iterations I have to do, I just choose the fast algorithm, but there are other that are quite low. Yeah, it takes more time, and but for today it's fine, the fast one. And then you have to prepare your MOFA. So you have to specify your model option that are all this one that is specified before, and also the training option. And then we run MOFA. It started a lot of iteration. Right now it was super fast. It saved all the information like in a H5 HDF5 object. And yeah, here have all the iteration. Yeah, for example, for right now, we choose like 3 factor. So it was like an easy way to see the situation because we have 3 input data and we just 3 factor. But we also can add the number of the factor, raise maybe to 20 or also 25, 30 or more, depends about your input data. And then here, for example, we have the variance explained for each factor. So we have factor 1, factor 2, factor 3, and then we have the image as an input, the RNA-seq and the methylation. And then you can see that, for example, the factor 1 contain more information expressed from the images. The factor 2 and the factor 3 are more from methylation. So from this kind of representation, seems that the RNA-seq data that we choose as an input are not so informative compared to the image and the methylation one. But maybe if we try to increase the number of the factor, maybe we can see different results. Yeah. And then here you can also plot the value, the latent factor across factor 1 and factor 2. Yeah. Yeah, for the code today is done. So, if you have any question or other stuff, or you can, these one are just an example for ovarian cancer, but on the TCGA, there are other 32 different tumor types that you can explore and maybe find the one that matches more the interest for your lab or your needs or collaboration. Yeah. Yeah.
What shape you style was like 1 million, so you just
Yeah, actually, because the embeddings are a lot, like 700. So the PCA was a kind of way to try to summarize the information contained in these embeddings. And just to summarize more the information. Yeah. Sorry, I cannot hear you. No, the number is established by the models, like the foundation model. So every time that you run again, you have this specific type because it's a pre-trained model that uses autoencoder and decoders. And so you cannot, one of the interesting about these embeddings that does one embedding has a particular meaning. For example, the embedding 345 doesn't mean something, or maybe just a random representation of the data, or maybe combination of different embeddings give us more information. So they are just right now open question in histopathological analysis, like digital pathology, just to, maybe next time that we see it, we have more information about that, but right now, there are more open questions. Yeah.
It's just for diagnostic images. Is there also plans or is there any part of this you do, for example, for the IHC, like for example, breast cancer you have for HER2 and
Yeah, actually, these HoverNet and ProfGigapath are specific for HND images, but there are other foundation model, convolution neural network that are maybe trained on that kind of images. And in that case, of course, you can rerun point pattern analysis or doing MOFA on this kind of output. Of course, you can do that. Yeah, this is just one of the possible examples. Yeah.
Done.
Okay.
Thank you so much. Thank you. Are you ready?
Thank you.
There are some difficulties, but this is always great.
Yeah.
Thanks a lot.
Thank you.